Um, so hello everyone. I am Susan. I'm from Reed College. I'm a senior majoring in neuroscience and I have a computer science minor. This summer I have this great honor to be a summer internship at Massachusetts engineer at Segre Lab. And today I'm going to talk about my summer research, some automation and optimization of a bioinformatics pipeline called Eclipser, which stands for Enrichment of Causal Loci and Identification of Pathogenic Cells in Single Cell Expression and Regulation Data. So first of all, I'm going to talk about a concept called genome-wide association studies, or GWAS. It explores which single nucleotide polymorphism are associated with specific trait or diseases. For instance, by comparing the genetic data of individuals with a specific disease to those in good health, we identify SNPs, so whether A or T or C or G, is showing significant difference in frequency in the disease cohort compared to the healthy group. When these differences reaches a statistical significance, we identify a strong association with a trait or disease. So for example, this is an illustration of a Manhattan plot depicting several strong associated risk loci. Each dot represents a SNP with the x-axis showing the genomic location and y-axis showing the association level. This example is taken from a GWAS study investigating coronary artery disease. So these peaks are indicating those genetic variants that are found more often in individuals with coronary artery disease. However, identifying these associations is only the beginning. To truly understand the mechanism of these polygenic diseases, we need to determine where and how these genetic differences affect genes and tissue or cellular function. The challenge is that most of the genetic associations are in non-coding regions, and that's we need to explore how certain SNPs may influence gene expression through regulatory mechanisms or affect gene splicing, leading to varied protein expression across different tissues and cell types, and eventually contributing to the complex disease. The complexity increases as each tissue might respond differently to those genetic variations. Therefore, although GWAS provide a broad overview, it does not pinpoint the specific tissue or possible or the exact possible causal mechanisms at play. It's also possible that a SNP strongly associated with the disease is merely in proximity to the actual causal variant and not the direct causes. So to address these challenges, our lab has developed a workflow that maps SNPs to potential causative genes. This includes integrating GWAS SNPs with non-expression and splicing quantitative trait loci, EQTLs and SQTLs, and testing if they're tagging the same causal variant. And thanks to the GTAX consortium, we now have the atlas of genetic regulation of gene expression in 49 health tissues. And thus we could calculate how specific loci is affecting gene expression and splicing events based on a linear regression model that controls for covariates. The Open Target Genetics database further aids this by employing a machine learning pipeline that predicts the most likely causal genes for over 300K GWAS variants of thousands of complex traits based on different functional genomics data, such as EQTLs, protein QTLs, epigenetic data, and deleterious protein coding variant. We then perform linkage disequilibrium clumping to consolidate proximal SNPs into single loci for analysis. With this refined data, we use single-cell RNA sequencing data to identify cell type-specific expression of all genes genome-wide, including genes mapped to GWAS loci. And then we test whether genes mapped to GWAS loci of a given complex trait are enriched for cell type specific expression in particular cell type compared to a null distribution. The leading genes contributing to the enrichment is also investigated. This helps us pinpoint the pathogenic cells and pathways involved in complex disease or traits. Ultimately, this comprehensive approach allows us to make informed judgment about potential causal variants, genes, and the specific tissue and cell types they affect, advancing our understanding of genetic diseases. However, this pipeline requires substantial manual manipulation, making it cumbersome to run on multiple traits and tissues. Therefore, my main project this summer has been to streamline this approach significantly and optimize the processes involved. This would facilitate running the analysis for various traits more efficiently and on single cell expression data from different reference tissues in HuBMAP with less manual effort. So to summarize, here are the main objectives for my summer research. First of all, to update the open target genetics table to include the predicted best guess genes for hundreds of thousands of GWAS loci associated with thousands of complex traits based on their machine learning algorithm. And second of all, to streamline the Eclipser pipeline to enhance its efficiency and usability. And last but not least, run Eclipser on vascular traits and arterial single nucleus RNA-seq data to identify pathogenic cell types and genes, providing crucial insights into vascular-related diseases. So for interacting with Open Target Genetics platform, I utilized their API via GraphQL. I built a query for each study ID contained in their database. Basically, you specify the data entries you want to include based on their provided format, and the corresponding information will be sent back to you. Due to server stability issues, I implemented checkpoints and timeouts to ensure robust data handling. The query retrieves information on the variant and the best locus to gene guesses, which are then stored in the data frame for further analysis. To streamline our bioinformatics pipeline, I use SnakeMake, which is a widely recognized Python-based tool for bioinformatics pipelines, which employs user-friendly rule definition to run scripts and manage dependencies seamlessly. So basically, SnakeMake builds a directed acyclic graph for the whole pipeline, with each node as a task, which could be a script you want to run or a pre-processing of data that you want to be done before running a script. And different tasks are connected based on dependencies. So once the pipeline is run, SnakeMake would automatically solve for those dependencies and run them in a sequential order. So to streamline the pipeline, I developed 2 operational modes for Eclipser to accommodate different user needs. So if you have a specific disease or trait that you're interested in, you could either search for the corresponding GWAS in the Open Target Genetics GWAS list, or you could specify which GWAS you want to use, whether it's publicly published or it's a result in your lab. And then those GWAS will be mapped to the genes based on EQTLs, SQTLs, and the Open Target Genetics machine learning algorithm. And then we would run clumping on those mapped genes. And then finally, you would input your single cell or single nuke RNA-seq data of the tissue of interest, and Eclipser would output the cell type specificity per GWAS loci. So I know in a lot of times running bioinformatics pipeline could be really annoying. So I automated this whole process so that users basically just need to input their directory of those required input files in the specific columns as instructed, and they could include a lot of parameters. For example, which column names correspond to the information that we want to extract, or the specific data type that's included in their GWAS table. And the whole pipeline would just run automatically and the corresponding output would be generated. So after automating this pipeline, I ran it on aorta artery with several vascular traits. This graph is showing a bubble map of enrichment of the corresponding GWAS for the specific cell types. So the color represents the significance of enrichment, and the size represents the fold change. So for example, you can see that here, coronary artery disease is found to be enriched in fibromyocyte and those 3 different kinds of vascular smooth muscle cells, and also one of the fibroblast cell types. Blood pressure is enriched in not only those cell types, but also another type of fibroblast. And migraine is only enriched for those 4 cell types. So we can sort of see a variation across these different GWAS loci. And as a negative control, IBD, which stands for inflammatory bowel disease, which is an immune disease, is only enriched in natural killer cells. Which makes sense since it's an immune disease and not of those vascular smooth muscles or ECs or macrophage. And another way of showing the result is through a forest plot in which different cell types are ranked by their significance for a specific trait or disease. And we can see that everything past this line is considered to be significant. So in conclusion, the project successfully increased the efficiency, flexibility, and usability of Eclipser. And for future directions, this pipeline could be applied to multiple traits, complex traits or diseases with available GWAS results and any single cell or single nucleus RNA-seq data to identify novel pathogenic cell groups. And for future, we plan to apply Eclipser to a range of vascular and other traits using available single cell or single nuke RNA-seq data on HuBMAP data port. So last but not least, I want to thank Segri Lab for providing this wonderful opportunity for me to learn. I really learned a lot in this summer. Special thanks to Eilid for your help and all the guidance. And also thanks to Gupta Lab, who provided data for us to analyze. Thanks HuBMAP for providing this invaluable opportunity, and thank my home institution, Reed College, for all the people who supported me to apply for this opportunity. And thank you. And does anyone have any questions?